Assalamualaikum and good morning. We are from Group 5 Biology Symposium, Johor Education College. We will do a presentation on cellular respiration.
I am the ALISA BALQISH, BINTI M.F.MOHD will discuss about glycolysis and degradation.
My name is Ahli Kumpulan. We will explain to you about Krebs Cycle.
I'm KIRTIGA and will explain about Untitled Chapter.
I am ISHAK. We will present about BINTI and Fermentation.
Hai, Assalamualaikum dan selamat sejahtera. Sekarang saya nak terangkan kepada anda tentang glycolysis. Okay, firstly I want to say I will explain about glycolysis and degradation in both language, English and Bahasa Melayu. So I hope you all can accept it. Okay, firstly, in glycolysis we need to set in our mind that glucose will split to pyruvate. Okay, this process will occur in cytosol. And before that, I want to give some short form for each process that we will learn in this topic. Okay, for pyruvate oxidation or linkation, the process will make pyruvate oxidize into acetate. Okay, for the Krebs cycle, electrons are removed from the acetate, and lastly, oxidative phosphorylation, the energy in electrons is used to make ATP. Also, each process will occur in specific place. For glycolysis and pyruvate oxidation or linkation will occur in cytosol, while Krebs cycle will occur in matrix of mitochondria, and oxidative phosphorylation will occur in inner membranes of mitochondria. Okay, this is an overview for glycolysis process. Now let us explore this process. Okay, firstly, we will have glucose, which has 6 carbon. Glucose is phosphorylated into glucose 6-phosphate. Okay, how glucose get that phosphate groups? It came from when ATP turns into ADP. So the ATP provide the phosphate group to glucose. This process of phosphorylation include the enzyme of hexokinase as catalyzed. Then glucose 6-phosphate will undergo isomerization and turns into fructose 6-phosphate. This is because glucose 6-phosphate is not stable. It's not a stable molecule, so it rearranges into fructose 6-phosphate. After we have fructose 6-phosphate, it will undergo phosphorylation again and turn into fructose 1,6-phosphate. Where does the another phosphate come? It comes from when ATP turns into ADP. It will donate phosphate group. In this phosphorylation, it needs enzyme phosphofructokinase as catalyzed. Fructose 1,6-phosphate will split and cleavage into 2 molecules. First is dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, 3GP. Okay, for your information, DHAP is not a stable molecule, so it needs to undergo isomerization into G3P. Okay, now best friend, when DHAP turn into G3P, so now we have 2 molecules of G3P. Okay, 2 molecules of G3P will undergo oxidation and phosphorylation and turns into 1,3-biphosphoglycerate, 1,3-BPG. Okay, this time you need to remember that G3P did not receive any phosphate group from ATP. It comes from inorganic phosphate in cytoplasm. After we have 2 molecules of 1,3-BPG that undergoes substrate level phosphorylation. Okay, I want to introduce you what is substrate level phosphorylation. Okay, process in which the phosphate group from substrate molecule is transferred into a molecule of ADP. It will form a new molecule of ATP. Only occur in glycolysis and in Krebs cycle. It will turn into two molecules of 3-phosphoglycerate. Okay, in this process, ADP will receive phosphate groups from 1,3-BPG to form a new 2 molecule of ATP. Okay, now as we all know that primary molecule is not stable as secondary and tertiary molecules, right? So 3-phosphoglycerate needs to undergo isomerization to turn into 2-phosphoglycerate. After we get 2 molecules of 2-phosphoglycerate, it will undergo dehydrogenation, which is this process will remove H2O and turns the 2-phosphoglycerate into 2 molecules of PEP. Lastly, 2 molecules of PEP will undergo again substrate level phosphorylation and turns into 2-pyruvate. The ADP will receive Pi, which is inorganic phosphate, and turns into 2 molecules of ATP. And for the results of glycolysis, we will have 2 molecules of pyruvate. Okay, this is the beautiful notes for you to jot down about glycolysis. Okay, now let's move on to link reaction or pyruvate oxidation. Okay, firstly, you need to set in your mind that this reaction links the glycolysis with the Krebs cycle. This reaction needs the presence of oxygen and occur in matrix of mitochondria. Why do link reaction needs the presence of oxygen? It is because it has a final electron acceptor at the end of reaction. This is an overview of link reaction for you to take a look. Easy words. Link reaction is a process of 2 molecules of pyruvate convert into acetyl coenzyme, which is need to undergo 3 process, which is we call D for decarbosylation, O for oxidation, and A is combination of coenzyme E process. Okay, now I want to make you more clear about the yield of the link reaction. Okay, for the decarbosylation process, 1 CO2 molecule per pyruvate will produce, which is just a waste product. For oxidation or dehydrogenation, which is just the same process, okay, 1 NADH per pyruvate will be used for oxidative phosphorylation, and then for the A combination of COA, 1 acetyl COA 2 carbon per pyruvate will be used in Krebs cycle.
Alright, so we will continue with Krebs cycle. Okay, so before I explain further about Krebs cycle, let's have a quick recap. Okay, so we have learned about glycolysis and link reaction. So in glycolysis, we produce 2 pyruvate, 2 net ATP, and 2 NADH and 2H+. Meanwhile, in link reaction, or the other name is oxidative decarbosylation, we will produce 2 acetyl-CoA, 2 CO2, and 2 NADH and 2H+. Okay, so in Krebs cycle, we will use the product in link reaction. Link reaction, the product that we use is 2-acetyl-CoA will be used in Krebs cycle. So, where the Krebs cycle occur? Krebs cycle occur in matrix of mitochondria. And how many steps are involved? It involves 8 steps. So this is Krebs cycle. In Krebs cycle, we use 2-acetyl-CoA to combine with oxaloacetate to form citrate 6 carbon. Okay, and then coenzyme A will be removed This is step 1. Okay, what is step 1? 2-acetyl-CoA combined with oxalic acetate to form citrate 6 carbon and coenzyme A will be removed. Okay, next, step 2. Step 2, citrate 6 carbon will undergo citrate 6 carbon will undergo isomerization or rearrangement to form isocitrate 6 carbon. Okay, so it will remove 2H2O and also will add 2H2O. That's what it means by isomerization. Maksudnya dia akan dia rearrange balik lah. 2H2O tu bila dah remove, nanti dia akan add balik H2O yang dia dah remove tadi untuk jadikan isocitrate 6 carbon. So isocitrate will form alpha ketoglutarate by oxidation and decarboxylation. This is step 3. Okay, oxidation maksudnya nanti electron carrier akan reduce, NAD+ akan reduce menjadi NADH plus H+. And decarboxylation means that carbon dioxide akan remove. Okay, so for the next step, step 4, alpha ketoglutarate 5 carbon will undergo oxidation and decarboxylation. Okay, so dia sama macam tadi. Maksudnya oxidation dia akan reduce NAD+ menjadi NADH campur H+, and decarboxylation maksudnya dia kurangkan C tu, jadi dibuang CO2, sebab tu jadi 5C ni jadi 4C. Okay, step 5. Succinyl-CoA dia akan menjadi succinate. Macam mana dia akan jadi? Dia akan converted succinyl-CoA converted to succinate. Macam mana dia converted? Sebab dia produce ATP by substrate level phosphorylation. Macam mana substrate level phosphorylation tu boleh produce ATP? Maksudnya nanti inorganic phosphate. Okay, so inorganic phosphate ni nanti dia akan campur dengan GDP menjadi GTP. And then GTP ni nanti dia akan pass phosphate yang ada kat GTP tu ke ADP. And then ADP dia akan form ATP. Okay, this is step 5. Okay, step 6, succinate 4C dia akan lupa yang ni, yang tadi NADH tu, sebab kita kan guna 2-acetyl-CoA, so nanti dia akan jadi dia akan jadi 2NADH 2H+, semua ni akan jadi 2. Nanti multiply dengan tu lah, sebab 2-acetyl-CoA kan, so maksudnya nanti jadi 2ADP 2ATP. Okay, so ni pun sama. Succinate kan nanti dia akan oxidize menjadi fumarate. So nanti electron carrier yang kita akan guna kat sini adalah FAD. FAD ni nanti dia akan menjadi FADH2. 2FAD akan menjadi FADH2. And then fumarate akan menjadi malate. This is step 7. Macam mana fumarate 4 carbon akan menjadi malate 4 carbon? Dia akan converted using water. And then the last step, last step, malik for carbon, dia akan mengalami oxidation. Dia akan oxidize menjadi oxaloacetate balik. Dia akan oxidize guna electron carrier NAD+ menjadi NADH plus H+. Okay. So, based on Krebs cycles ni, produk yang kita akan dapat adalah 4CO2, 6 NADH plus H+, 2 FADH2. And then macam mana kita nak ingat dia punya step? So kita boleh guna akronim. Akronim apa kita nak guna? Boleh guna je, kalau saya guna ni. Can I keep some succinate for myself? O tu untuk oxaloacetate. C untuk citrate. I untuk isocitrate. K untuk alpha ketoglutarate. S for succinyl-CoA. Succinate for succinate. And F for fumarate. And M for malate. Okay, that's all from me. Thank you.
NADH synthesized in glycolysis can enter mitochondria. Why is that so? It is because mitochondria membrane is not permeable to NADH. So that the electrons from NADH in cytoplasm must be transferred to a molecule that can freely move through mitochondrial membrane. In malate shuttle, oxaloacetic acid in mitochondria moves into cytoplasm and receives electron from NADH. Oxaloacetic acid is reduced to malate and moves into mitochondria. NADH is oxidized into NAD+ and remain in cytoplasm to receive electrons from glucose during glycolysis.
Hi and Assalamualaikum, I am Aisyah. I'm going to share about fermentation, the last subtopic in cellular respiration, chapter 5. We'll start with fermentation. It will occur during the absence of oxygen and occur in cytoplasm. Fermentation will produce ATP, but ATP produced in anaerobic is lesser than ATP produced in aerobic. Aerobic means this fermentation, And then this fermentation process will involve glycolysis and reduction of pyruvate by NADH. If the question is asking about role of NAD+ and role of NADH, well, the role of NAD+ as oxidizing agent, the role of NADH as reducing agent. Fermentation can be divided into two types, which is lactate fermentation and alcohol fermentation. For lactate fermentation, it will produce ATP and lactate in animal or specifically in muscle cell. The acetaldehyde as final electron receptor, this is a general lactate fermentation equation, starts with glucose converted into 2 lactic acid and 2 ATP molecule. Secondly, alcohol fermentation will produce ATP, carbon dioxide and ethanol in plant. The final electron receptor is pyruvate, different from the lactate fermentation, which is acetaldehyde as final electron receptor. This is the general equation for alcohol fermentation. Starts with glucose, converted into 2 ethanol, 2 carbon dioxide molecule and 2 ATP molecule. Well, if we are talking about a process, of course it has its own importance. So we will discuss about importance of fermentation. Can be divided into 3 types, which is alcohol fermentation, involved in bakery, which is dough, rice, vinegar, beverages in wine and alcohol production. Next is lactate fermentation, involved in dairy product, for example for cheese and yogurt production. And lastly, for local example, which is tempeh, fermented soybean, and chinchalo, fermented shrimp. Now we'll start with lactate fermentation. Lactate fermentation will occur in muscle cell. Aerobic respiration switch to lactic acid fermentation when oxygen is insufficient. So the importance of lactate fermentation is ensure ATP production continues in absence of oxygen. This is the simplified diagram for you to understand lactate fermentation. We'll start with glucose. Glucose will undergo oxidation to pyruvate through glycolysis, and then 2-pyruvate will be reduced to 2-lactic acid. So where did ATP and NADH come from? So ATP and NADH will come from substrate level phosphorylation. These both ATP and NADH are produced from substrate level phosphorylation. Okay, and then we have another information, which is the enzyme that convert pyruvate into lactate acid is lactate dehydrogenase. So lactate dehydrogenase will convert pyruvate into lactic acid or lactate. So this is the description about the diagram above. So first is reduction. Pyruvate reduced by NADH to produce lactate. So pyruvate is the pyruvate will receive electron from NADH. So that's why we call pyruvate as the last or as the final electron acceptor for lactate fermentation. And then we have NADH is oxidized to NAD+, and then this regenerates supply of NAD+ needed for glycolysis continuation. So NADH that oxidized into NAD+, then NAD+ will be used for glycolysis process. So this process will be continue. So this is the general equation for lactate fermentation. Well, the final product is lactic acid, right? So if we have too much lactic acid amount in our body, it is not good, because when lactate accumulates, but it is gradually carried to liver, converted back to pyruvate when oxygen is available. This is to ensure lactate removed from muscle cell, as we know that lactate fermentation is occur in muscle cell. So accumulation of lactic acid causes fatigue, cramp, lower blood pH in muscle. So that's why we cannot have so many or too much amount of lactic acid in our muscle cell, because it has its own effects. Secondly, we have alcohol fermentation. This is the simplified diagram for alcohol fermentation. Same as the lactate fermentation. Starts with glucose. Glucose will be oxidized to pyruvate through glycolysis. Okay, and then pyruvate will undergo decarboxylation as the first process. Okay, so this is the first process, like this one, decarboxylation, which is removal or release of carbon dioxide, this one, and pyruvate converted to acetaldehyde. And then the second process is reduction. Reduction occurs here. This is number 2. In process number 2, acetaldehyde is reduced by NADH to ethanol, reduced, and acetaldehyde is the final electron acceptor. It will accept electron from NADH NADH. So that's why we call acetaldehyde as the final electron acceptor. Like the lactate, pyruvate is the final acceptor, while in alcohol fermentation, the acetaldehyde is the final electron acceptor. So lastly, this regenerates supply of NAD+ needed for the continuation of glycolysis process. So this thing will be continued like before, like the lactate fermentation, and NAD+ will undergo oxidation to form NAD+, and NAD+ will undergo reduction to NADH. The last thing is, this is the essay example for this subtopic. So the question is, describe alcohol and lactate fermentation. You can take these points and insert in your essay question. Thank you.